Welcome to the Deep Dive, the show where we really try to cut through the noise and get straight to what matters,
Yeah, there's just so much information out there, isn't there? It's hard to know what's truly important.
especially about ourselves. So we're here to give you that shortcut, distill these big topics into clear, maybe surprising insights.
Exactly. We take a pile of research, articles, maybe lectures, and pull out the key bits. Hopefully you walk away knowing more, maybe with a few aha moments.
And today we're diving into, well, a really fascinating part of the brain, the limbic system.
The emotional core often gets a bit overlooked, doesn't it?
It really does. You know, in med school, prep or whatever, they might gloss over it, focus on spinal cords, bladder control, more practical things maybe.
Right, but we think it's one of the most, well, wonderful, interesting parts of the whole brain.
Absolutely. We've been digging into this great Stanford University lecture, really trying to unpack emotion, memory, behavior,
Yeah.
all linked to this system. So let's get into it.
Okay, so where do we start? Its origins, maybe?
Yeah, the first name wasn't even limbic system, was it?
No, not at all. Early on, especially looking at rodents, they called it the rhinencephalon. Literally, nose brain.
Nose brain, why?
Because in a rat, the olfactory parts, the bulb, the projections, they're huge. Like 40% of their whole brain is dedicated to smell. So early researchers saw this massive structure and thought, well, it must be all about smell.
Okay, that makes sense. Purely sensory input initially.
Pretty much. But then things got complicated.
How so?
Well, as the 20th century went on, scientists started seeing the same area was involved in, emotions, fear, aggression, mating.
So then you have a conflict. Is it the nose brain or the emotion brain?
Exactly. The lecture called it a central conflict. Do we stick with rhinencephalon or switch to limbic system? It sounds like just semantics, but it was about a fundamental shift in understanding.
So how do they sort it out?
It came down to thinking like an ethologist. Someone who studies animal behavior in the wild.
Right, looking at the animal's actual life.
Precisely. For a rat, its whole emotional world is smell. Fear is triggered by predator smells, mating by pheromones, food by scent.
So for the rat, the nose brain is the emotional brain. They're inseparable.
You got it. The conflict dissolves when you look at it from the animal's perspective.
Okay, that clears up the rat situation. But what about other animals? Or us? Our emotional lives aren't exactly smell-driven.
Great question, and that's the key insight. It's not just about smell universally. Look at birds. Their limbic systems get huge inputs from auditory areas. Birdsong, right. Crucial for their social lives.
So it processes whatever sense is most relevant emotionally for that species.
Exactly. Or electric fish. Their limbic systems are geared towards electroreception for social stuff. So the big idea is the limbic system processes the sensory information most pertinent to a species' emotional life, whatever drives their world.
That's a much broader, more powerful idea than just nose brain. Okay, so we've got this specialized emotional processing hub. How does it fit into the bigger picture of the brain?
Well, a really helpful, though simplified model came about in the 1950s from Paul MacLean. His triune brain idea.
Triune, like 3 parts.
Yeah, 3 layers, basically.
Ugh.
At the core, you have the oldest part, the reptilian core. It's phylogenetically conserved, meaning like lizards, birds, mammals. We all have this basic setup.
And what's in there? Hypothalamus, pituitary, brainstem.
Yeah, and the midbrain. And its job is, well, kind of the boring stuff. Automatic regulation, body temperature, blood sugar, blood pressure, heart rate.
Vital, but not exactly thrilling.
Sounds boring, yeah, until it breaks. There's this terrifying condition called Ondine's curse.
Ondine's curse. What's that?
It's incredibly rare, thankfully. Damage to the midbrain stops automatic breathing.
Stops. Automatic breathing. So you have to consciously remember to breathe.
Exactly. And when you sleep, you stop breathing. But you don't just suffocate peacefully. The CO2 builds up, jolts you awake, gasping. People with this condition often die not from lack of oxygen, but from sheer sleep deprivation.
Wow. Okay, suddenly that boring core seems pretty crucial.
Doesn't it just? So that's layer 1. Layer 2, sitting on top, is the limbic system itself. MacLean saw this as mostly a mammalian thing, where emotions really get complex.
And layer 3?
That's the cortex, the gleaming analytical machine, especially big in primates and us. Higher thought cognition.
Right. But didn't you mention Descartes earlier? This idea that the thinking cortex is totally separate from the emotional limbic system? That's wrong.
Completely wrong. That's Antonio Damasio's whole point in Descartes' error, this idea of pure, detached reason. He calls it complete nonsense.
So how do they interact? Can you give an example?
Oh, definitely. Ever made a really, let's say, questionable decision when you were super angry or stressed?
Maybe once or twice.
Right. That's your limbic system flooding your cortex, influencing your rational thought. But it goes the other way, too.
The cortex influences the limbic system.
Absolutely. Just thinking about something stressful, an exam, public speaking, even just, mortality can trigger the release of stress hormones like CRH from deeper brain areas.
So there's constant back and forth, tons and tons of crosstalk, as the lecture put it.
Exactly. They're deeply intertwined.
Okay, let's zoom back into the limbic system itself. Who figured out how its parts connect?
A key figure was James Papez back in the 1930s. He was really the first to see these structures not just as random bits, but as an organized module. He mapped out the Papez circuit.
The core emotional loop.
Kind of, yeah. He established the limbic system as the emotional part of the brain systematically.
And when neuroscientists talk about connections, they use terms like nuclei and projections.
Right. Nuclei are just clusters of nerve cell bodies, gray matter. Projections are the bundles of axons connecting them, the wiring, basically. Often myelinated, white matter for speed. Papez was tracing these wires.
Is there like a simple rule for how this wiring works? A logic to it?
There's a really neat unifying principle. Pretty much every part of the limbic system deep down wants to tell the hypothalamus what to do.
The hypothalamus again, from the reptilian core.
Yep. It's a central hub connecting emotions to the body hormone release via the neuroendocrine system and the autonomic nervous system, like fight or flight.
So all these limbic areas are vying for the hypothalamus's attention.
Kind of. They stimulate it, but they also inhibit each other to try and get their message through to control the body's response.
And how does that work? Is there a way one part gets more influence than another?
There's a basic neurobiological rule. The fewer synapses, the fewer connection points between a limbic part and the hypothalamus, the faster and stronger its influence.
Fewer connections equals more power.
Essentially, yeah. Being just one synapse away gives you a lot more control.
And this connects back to smell, doesn't it?
Amazingly, yes. Most senses, vision, hearing take like 3 or 4 synapses to really hit the emotional circuits hard, but olfaction, smell,
How many?
1 synapse. A straight shot to parts of the limbic system.
Wow. So even though our sense of smell isn't dominant like a rat's, that direct wiring explains why a smell can just instantly zap you back to an old memory or feeling?
Exactly. It bypasses a lot of the usual processing. It's a direct line.
That's fascinating. And sometimes the wiring itself is weird, inefficient.
Oh, totally. The lecture mentioned the stria terminalis. It's a path from the amygdala, we'll get to that, to the hypothalamus. Logically, they're right next door. It should be a short hop.
But it isn't.
Nope. It takes this huge loopy detour around the hippocampus. Really nutty, inefficient wiring.
Why would it do that?
Stuff like that often reflects how the brain developed embryonically, or its evolutionary history. It points to how evolution is not an inventor. Evolution is a tinkerer.
A tinkerer.
Yeah, it doesn't design things perfectly from scratch. It modifies what's already there. Like taking crude motor systems for claws and adding an extra pyramidal system in primates for fine finger movements, good enough for piano trills. Even if the underlying wiring isn't the most direct route, it's patched together.
Okay, evolution the tinkerer. Got it. So let's meet the key players in this limbic system. Who are the main structures?
All right, first pass, knowing it's all interconnected. Let's start with the amygdala.
The almond-shaped one.
That's it. Famous for fear, anxiety, aggression. But interestingly, also involved in male sexual motivation.
Aggression and sexual motivation linked.
The source suggested that overlap might, in some pathological cases, help explain why those behaviors can get confused or intertwined.
Okay, and what counterbalances the amygdala's aggression role?
That would be the septum. It seems to play an opposite role, inhibiting aggression. You see this a lot, cross-inhibitory projections, where areas keep each other in check.
Makes sense. What about the hippocampus? I know that one memory, right?
Definitely famous for learning and memory, but it's not just memory. Crucially, it also helps turn off the stress response.
How does it do that?
It monitors levels of stress hormones, glucocorticoids. It's like a feedback sensor. If levels are high, it signals back to, guess where?
The hypothalamus.
Bingo. Tells it to calm down the stress signal, so there's a logical link. Memory, learning what's dangerous or safe, helps regulate future stress responses.
Very cool. What else? Mammillary bodies.
Relevant to maternal behavior, yeah. But maybe more central for things like depression and addiction are the VTA and the nucleus accumbens.
VTA, ventral tegmental area.
Right, and ANAC, These are packed with dopamine neurons, key players in the reward system.
So dopamine equals pleasure.
Ah, that's the old view.
Mm-hmm.
The lecture really emphasized a huge shift here. It's not about feeling pleasure, it's about anticipating pleasure.
Anticipating it.
Yeah, and crucially, it's about powering the behavior you need to do in order to get the reward. It's the motivation, the seeking.
Can you give an example?
Sure. The classic monkey experiment. Train a monkey to press a lever when a light comes on to get juice. At first, dopamine spikes when the juice arrives.
The reward itself.
Right, but very quickly the dopamine spike shifts. It starts firing when the light comes on the signal, the cue, not when the juice arrives.
So it's driving the lever pressing. Proceeds.
Exactly. It's the pursuit of pleasure, the wanting, the seeking. And that's far more relevant to understanding addiction than just the momentary high.
Wow, okay. That changes how you think about reward. Now, the prefrontal cortex. You said it was controversial to include it.
Yeah, Volina, this neuroanatomist, argued it belonged with the limbic system based on connections, even though it's cortex. And functionally. He was spot on. The PFC does all sorts of utterly uncortex-like things.
Like what?
Emotional regulation, impulse control, long-term planning, delaying gratification. All the stuff that separates us from, well, pure reaction. The lecturer called it, without question, the most interesting part of the brain.
And it's uniquely human, in a way.
Disproportionately huge in humans, yes. And fascinatingly, it's the last part of the brain to fully mature. Full myelination might not finish until your mid-20s.
Mid-20s. That explains a lot about teenagers.
Yeah. The lecturer specifically mentioned impulse control, and it's not working yet very well, most of you guys. A direct neurobiological underpinning for adolescent behavior.
So why did it get so big in humans?
Well, Ian Dunbar's social brain hypothesis is compelling. He looked at 150 primate species and found the best predictor of PFC size was average social group size.
So it evolved for dealing with other primates.
Essentially, yeah, for gossip and social relations and appropriate behavior and social intelligence.
Sure.
Managing complex social landscapes.
And there are specific parts within the PFC, like the anterior cingulum.
Hey, a subregion, it lights up with empathy, like if you see someone poke a loved one's finger. And its dysfunction in depression points to maybe a pathological hypersensitivity to the pains of life.
Heavy stuff. Okay, any other key nuclei, maybe in the hypothalamus, we should quickly mention?
Just briefly, the ventromedial hypothalamus and medial preoptic area are involved in sexual behavior. There are observed sex differences in size, and some landmark studies linked size differences here to sexual orientation and even transgender identity, the wrong body idea. And the paraventricular nucleus, the PVN, that's the starting gun for the stress response. It pumps out CRH, gets signals from everywhere, a stub toe, thinking about world suffering. It integrates it all to kick off that stress cascade.
Wow, so many interconnected parts. How on earth do scientists study all this?
Yeah.
How do they map these emotional circuits?
It's a whole toolkit. One classic way is studying lesions damage, sometimes accidental, like, war injuries, or famous cases like patient HM who lost his hippocampus.
Right, taught us about memory.
Yeah.
Or that weird fencing foil injury.
Yeah, patient NA. Miniature foil through the nose into the septum gave insights into aggression and inhibition. Or they create intentional lesions in animal models. The basic idea is break it, see what stops working.
Okay, lesions. What else?
Stimulation zapped neurons with an electrode, see what happens. Recording listen in on electrical activity, even down to single neurons or ion channels. Anatomy just meticulously tracing the wires of the circuit.
And more modern techniques.
Biochemical methods measuring neurotransmitters, hormones, molecular biology looking at gene expression, and of course, human brain imaging.
CT scans, MRI.
Right, and functional imaging, looking at metabolic activity. That's how we know, say, the amygdala can get bigger and hyperreactive in PTSD, or the hippocampus can shrink in severe depression, shows the brain's plasticity.
But interpreting all this data for emotions must be way harder than for, say, mapping motor control.
Oh, a gazillion times harder, yeah. Huge caveats.
Like what are the pitfalls?
Well, one big one is the centers versus fibers of passage problem.
Centers versus.
Imagine you want to find where garlic comes from. You see trucks on Highway 101 carrying garlic. So you bomb 101, no more garlic arrives. Do you conclude 101 is the garlic production center?
No, you just destroyed the transport route.
Exactly. Many brain areas are both processing centers and major highways for signals passing through. A lesion might disrupt function just because it cut a crucial cable, not because it destroyed the center itself. Makes interpreting lesions really tricky.
So calling something the fear center or the aggression center is too simple.
Way too simple for the limbic system, where emotion, sensation, memory are all mixed up, which leads to the other huge caveat. You gotta know your species. Ethology is critical.
How so?
Stimulate the same spot in the amygdala. A lion might extend its claws. A human might just curse, say shit.
Different behaviors?
But both expressing irritation, right? Just in species-specific ways. Or stimulate a maternal area. A rat might gather newspaper for a nest. A monkey might cradle a soft, cylindrical object. Both maternal, but look totally different.
You need that behavioral context.
Desperately. There was this classic mistake: stimulating a rat's hypothalamus made it attack a mouse. Researchers thought, aha! Aggression center.
What?
They stimulated the same spot in humans. Did they get aggressive? No. They suddenly got hungry, ripped open a cereal box, and started eating. Why?
Because rats eat mice.
Exactly. It wasn't an aggression center. It was a predatory feeding behavior center. Totally different interpretation once you know the species' natural behavior.
Wow. Okay, know your species. Anything else?
Know your individual, too. Stimulate that aggression spot in a dominant baboon, you might get a threat display. Do it in a low-ranking baboon in the same group.
Nothing happens.
Probably nothing, because expressing aggression in that social context is tightly inhibited for that individual. Context, species, individual, it all matters.
Okay, this is complex. Now, we've mostly talked about the brain influencing the body, but you mentioned feedback. The body talking back to the brain.
Yes, hugely important. How stuff going on in the body will influence limbic function. Which brings us to the James Lange theory of emotion.
Ah, yes, the one you said sounded ridiculous.
Haha, yeah, from the early 1900s. It sounded totally backwards, but turns out there's a lot of truth to it.
So, remind me, what's the core idea?
Instead of, brain feels fear, body reacts, heart pounds. James Lange flips it. See, bare body reacts first, heart pounds, breathing quickens, muscles tense. Brain reads these bodily signals, then decides, aha, I must be feeling fear.
So the feeling follows the physical reaction. We're scared because we run.
That's the gist. The brain interprets its own body state to determine the emotion.
Okay, that does sound counterintuitive. What's the evidence?
Well, quite a bit, actually. Remember Stanley Schachter's epinephrine study from the 60s?
Vaguely. Adrenaline.
Right. He gave subjects shots of adrenaline epinephrine, basically mimicking physical arousal, but they didn't know what they got. Then they interacted with someone acting either super happy or super angry.
And what happened? Did the adrenaline make them feel a specific emotion?
No, that's the key. It didn't cause an emotion, but it magnified whatever emotion was suggested by the social situation. If the Confederate was angry, the adrenaline subjects felt more angry. If happy, more euphoric.
So the bodily state modulated or amplified the feeling.
Exactly. It jacked up the intensity. The body state didn't dictate which emotion, but how strongly it was felt.
Interesting. What else supports this?
Think about Valium or other benzodiazepines. They're anti-anxiety drugs, but they're also muscle relaxants. Part of anxiety is monitoring your own body tension. If the drug relaxes your muscles, that feedback loop is broken. Your brain gets the signal, muscles relaxed, and interprets it as, so maybe things aren't so bad. It reduces the feeling of anxiety partly by changing the physical state.
Like biofeedback or meditation too? Learning to control breathing or heart rate can calm your mind.
Precisely. You're using the body-to-brain pathway. Learning to regulate the physical state directly impacts the emotional state.
Okay, give me a really relatable example.
The post-argument loop. You have a big fight, you resolve the issue fairly quickly in your head, but your sympathetic nervous system heart rate, adrenaline, takes minutes to return to normal.
Right, you still feel agitated even if you've made up.
Exactly, and because you still feel physically keyed up, your brain tries to explain it. Why am I still feeling like this? It must be for some reason. And then you might start dredging up old issues. It's because of what they did in 1968. The lingering physical state fuels the emotional reevaluation.
I think we've all been there. And you mentioned gender differences here.
Yeah, the lecture dropped a factoid suggesting females, on average, might take a bit longer physiologically to return to baseline after strong sympathetic arousal or even after orgasm, which might humorously explain some common post-stress or post-intimacy dynamics.
Like one person wanting noodles while the other wants to cuddle and process.
No, something like that. The underlying physiological recovery time differs, leading to different immediate feelings or needs, potentially. Again, body-influencing feeling.
One less piece of evidence.
Just try it. Force yourself to smile, like really broadly, for 30 seconds. It often genuinely lifts your mood a bit. Or studies showing that sitting up straight after getting good news makes you feel prouder and happier than if you slump.
Posture affecting emotion. Wow. The body really does talk back.
It absolutely does. It's constant dialogue.
So wrapping this all up, what's the big picture takeaway from this deep dive into the limbic system?
I think it's really seeing the brain, especially the emotional brain, not as some isolated command center, but as part of this deeply integrated two-way system with the body. The limbic system influences physiology, sure, but physiology profoundly influences the limbic system right back.
Which makes sense of things like the Twinkie defense,
Yeah.
right? The idea that what you eat could affect your mood or behavior, that's the body-to-brain loop again.
Exactly. It highlights that other half of the equation.
Right.
And hopefully, going through all this, the anatomy, the ethology, the psychology, the physiology, gives you the sort of buckets necessary to understand how all these different approaches are needed to tackle complex behavior.
It's not just one field's problem. You need all those perspectives.
Absolutely.
So a final thought for our listeners to ponder.
Well, given how much our bodily state seems to influence our feelings, the whole James Lange idea made me reflect on this. How much of what you feel day to day is actually your brain interpreting subtle signals from your body? Your posture, your breathing, your gut. And if that's true, what does that mean for self-awareness? Could paying closer attention to those